Cellular migration is a critical wound healing process that can drastically alter the outcome of a healing wound. This protocol allows researchers to better understand the effect of various compounds on cellular migration during wound healing, which can oftentimes lead to targeted treatment strategies for clinical medicine. The scratch assay is a cost-effective and high-throughput approach for generating meaningful wound healing data prior to costly and time-intensive in vivo testing.
The scratch assay as a method is particularly relevant in wound healing applications, most specifically the skin. However, the assay is really a migration assay and could be applied to any cell type where migration of cells is of interest to measure or to capture. Similar to classic cell and tissue culture, standard operating procedures, training, technique, and practice. Those are key to success with the scratch assay. Demonstrating the procedure will be Nathan Cruz, a master's student, and Oscar Lujan, an undergraduate researcher.
To begin this procedure, prepare the Biosafety 2 cabinet using a 70% isopropyl aqueous solution to perform a sanitation wipe, beginning at the back and side walls, and working down to the base plate. Also wipe down all materials that will be used in the assay before placing them in the cabinet. Next, obtain vials containing the desired cryopreserved cell line and place one in a 1 cm deep water bath at 37 degrees Celsius to thaw. Add 10 milliliters of medium supplemented with 10% FBS into a T75 tissue culture flask. Open the vial containing the cell stock and carefully aspirate the solution. Then, transfer the cells into the T75 flask.